Hello, my name is Rahi Shah. I'm from the elucidate lab, and the project that I'm working on is about NMUR1, which is a neuropeptide Neuromedin U receptor, and how it maintains insulin signaling homeostasis in C. elegans. C. elegans is a great model organism for my project because I'm looking at the expression patterns of ILPs, or insulin-like peptides, in different neurons. And because C. elegans has a transparent skin, it allows me to do so. Firstly, to provide a bit of background information, one of the current lab members, Dennis Sifoglu, has found that NMUR1, which again is a neuropeptide receptor, has opposing effects on the longevity of C. elegans, and these effects depend upon the activity of DAF2. DAF2 is an insulin receptor to which some of the ILPs, or insulin-like peptides, bind to as ligands. So, he proposed that when DAF2 activity is high, NMUR1 works to inhibit it, and on the other hand, if DAF2 activity is low, NMUR1 promotes it. These differing responses work in order to maintain optimal level of pharyngeal health and survival in C. elegans. I am testing the hypotheses that if this effect is actually happening at the level of ILPs. C. elegans have 40 ILPs, and previous literature has already shown that these ILPs can act as both agonists or antagonists, depending upon the internal and the external environment of the worms. I started testing this hypothesis by looking at DAF28, which is an ILP that is expressed in at least 4 pairs of neurons, including 2 longevity-regulating ones, ASI and ASJ. I created a homozygous NMUR1 mutant strain, along with a homozygous transgenic insertion of DAF28 promoter linked to the mCherry gene, which allows me to visualize changes in expression patterns. This data shown here includes the quantification of the DAF28 intensity in ASI, which is a longevity inhibiting neuron. I found there to be no significant differences in the expression of DAF28 between wild-type and NMUR1 in ASI, as shown by the p-value here. However, what about the other DAF28 expressing neurons? To explore this question further, I will be looking at ASJ, which is a longevity promoting neuron, and also at ADL. This is an image from one of my analysis where the ASI neuron is being lit up in red, which is right next to ADL. If there is a difference in expression observed between the wild type and the NMUR1 mutants, it will prove the hypothesis correct that NMUR1 does indeed act through ILPs to regulate the activity of DAF2. Thank you.